So we continue our drill about the metabolism of amino acids, and I think with the previous video we kind of finished the general metabolism of amino acids. So now we are starting to deal with the specific amino acids and their metabolism. So in the previous few videos, we had seen about what generally happens with the amino acids as far as their metabolism is concerned. Now we will be seeing as to the individual amino acids and how their metabolism actually is carried on. But before we go on to that, I would like to explain an extremely important concept about the classification of amino acids as you can already see, that is glucogenic and ketogenic amino acids. This is very important, and this is something that is very pertinent to our discussion on the specific amino acids. Now, what is all this? Let me tell you. In the previous few videos, we have actually explained as to what happens to the nitrogenous part of the amino acid, right? We have shown, or rather we have seen, that the amino group, the amino part of the amino acids, that is actually transferred, and then it is released in the form of ammonia, and then ammonia being toxic for the body, it is converted to a less toxic intermediate, that is urea, via the urea cycle, and is excreted off, right? So we have completed with that part. But the question remains as to what happens to the carbon skeleton of the amino acid. The nitrogen part is taken care of, but what of the rest of the part? The rest of the part, that is the carbon part, what happens to that? That is where the specific amino acid metabolism comes to play, and we will be studying about that. What we see is that after the metabolism of the carbon skeleton that is left, all the 20 amino acids that are present in our body, they ultimately form one of these 7 metabolites. One of these 7 metabolites, right? So after the ultimate metabolism has taken place of the carbon skeleton of the amino acids, it has been seen that all the 20 amino acids present in the body ultimately form one of these 7 things, right? Now, very amazingly enough, all these 7 are linked together in a cycle. All these 7 are linked together in a cycle, which is Krebs cycle or the citrate cycle, right? So why am I telling this? I am telling this because this information is pertinent to the classification of amino acids as to whether they are glucogenic or ketogenic. Let me explain. You see, oxaloacetic acid, this is abbreviated form, by the process of gluconeogenesis, it can form glucose, right? Also, pyruvate in the same process of gluconeogenesis, by the help of the enzyme pyruvate carboxylase, it can form oxaloacetic acid. Now, why is oxaloacetic acid so important? It is important obviously because it is the link between gluconeogenesis and this Krebs cycle. It is also important because all amino acids which lead to the formation of oxaloacetic acid directly or indirectly will lead to formation of glucose by gluconeogenesis. So all those amino acids which ultimately lead to the formation of oxaloacetic acid, and which in turn, by the process of gluconeogenesis, forms glucose. All these amino acids are called glucogenic amino acids. Alright, so the requisite is that ultimately, directly or indirectly, but ultimately, oxaloacetic acid has to be formed. Alright, so it can be that the amino acid, for example tryptophan or alanine or cysteine, they are forming pyruvate at the end. But pyruvate in turn, by the enzyme pyruvate carboxylase, can convert into oxaloacetate, and that by the process of gluconeogenesis can form glucose. So these amino acids are all glucogenic amino acids. Alright. Then what are ketogenic amino acids? You see, ketogenic amino acids are those amino acids which ultimately lead to the formation of this acetyl-CoA or even acetoacetic acid, or some say it is to be acetoacetyl-CoA also. Alright, acetoacetic acid or acetoacetyl-CoA. Right, these amino acids are ketogenic. What does that mean and why? Let me explain. You see, acetoacetic acid itself is a ketone body. So all those amino acids which lead to its formation directly or indirectly via acetyl-CoA are all ketogenic amino acids. Alright, they are all ketogenic amino acids. But a question arises. Acetyl-CoA can also ultimately lead to the formation of oxaloacetic acid, that to glucose. So why are these amino acids not glucogenic as well? Very simple answer. You see, from acetyl-CoA we cannot get oxaloacetic acid without first using up a molecule of oxaloacetic acid to form a molecule of citrate. That is the reason I have actually listed citrate in this cycle. Are you getting me? You have to use a molecule of oxaloacetic acid with acetyl-CoA to form citrate, and then the cycle will continue. So if acetyl-CoA is being formed by these amino acids, it means that at the end they are not giving rise to oxaloacetic acid, because at the beginning itself one molecule is used, at the end you're getting one molecule, so 1 minus 1 is 0, so no oxaloacetic acid molecule is formed, and hence glucose is not formed ultimately by these amino acids. Hence they are what we say to be ketogenic amino acids. So I actually wrote down this list at first, because obviously I do not remember all these amino acid names, so I had to jot them down from the internet and list them here. But what we can say here, that there are some stars listed besides some amino acids here, as you can see already. If I think this is legible, so there are some stars given here. What are the star marks for? Let me explain. The black stars here, you see, tryptophan, isoleucine, phenylalanine. What does this mean? The black star is actually corresponding to those amino acids which are both glucogenic and ketogenic. I have abbreviated, obviously. They are both glucogenic and ketogenic. See, all those amino acids: tryptophan, phenylalanine, tyrosine, and isoleucine. These are the 4 amino acids which are both ketogenic and glucogenic. And obviously the red ink here is corresponding to the ketogenic amino acids, and the blue ink is corresponding to the glucogenic ones. Now I think there are also some amino acids like leucine with a red star. So what does that imply? The red star implies only ketogenic. All right, only ketogenic. So that we have for leucine here and lysine here. So leucine and lysine are the only two amino acids which are only ketogenic. They are not both glucogenic and both ketogenic, right? The rest of the amino acids, which account for about 14 amino acids, we have 4 amino acids which are both glucogenic and both ketogenic, and 2 which are only ketogenic. The rest of the 14 amino acids are only glucogenic, and obviously I have not marked them because 14 is a big number, so you can make that out. But that is not the important thing here. The important thing is not to, I feel like it's not to remember which one is which one. It is to remember what stands for what. Like, what is glucogenic? What do you mean by that concept? This concept is important rather than these jotted down examples. That is what I feel like. Because when we will be going into the specifics of this amino acid metabolism, we will be delving into how these things are happening. Like how these amino acids are forming acetoacetic acid by one pathway, and these amino acids are forming pyruvate, how they are forming oxaloacetate. These arrow marks are basically what we are going to study, how this is happening. But we have studied the classification of amino acids on the basis of what they are forming at the end, or rather, to be more precise, what their carbon skeleton is forming at the end, right? So that is an important concept, and that is something that I felt like should be discussed at the beginning. That is glucogenic and ketogenic amino acids. All right. Now, we will be seeing as to the specific amino acid metabolism, and the first amino acid that we will actually be discussing is glycine, actually. So we start with glycine. All right. So, all right, here's the thing. Any amino acid metabolism that we study, we first have to see how that amino acid is actually formed. That is, the different reactions, the different pathways by which that particular amino acid is being formed. Then secondly, we have to see how this amino acid is broken down, or how this amino acid is forming different products by the different metabolic functions of this amino acid. All right. So two things. First is obviously formation. All right. So let's talk about how glycine is formed. All right. Now, obviously, there are an array of reactions by which glycine is formed. So let's talk about them one by one. The first amino acid which I want you to remember, it's very important. The first one is very important. First one that I want you to remember is glycine's formation from the amino acid serine. All right. So what is the reaction? All right, here it is. You see, serine can form glycine, and this is a reversible reaction. All right, this is the reaction. This is typically the reaction, but obviously this is not finished. What is happening in this reaction? For knowing what is happening in this reaction, we need to kind of, just have a little bit idea about what the molecular structure are of serine and glycine. Glycine is very, very easy. It's like, it's nothing. It's the easiest, it's the simplest amino acid. So it has 3 of these things, carbons. One is obviously carboxyl end. All right. Oh, did I say it has 3? Oh my god, no. It has 2. It has 2 actually. I'm sorry. I'm very sorry. This, and in this end, it has the amino. So this is the zwitterionic form of glycine. I'm very sorry, I gave 3 carbons, I'm sorry. So it has 2 carbons and 1 carbon is actually linked to the carboxyl end and the amino end as well, and there is nothing else. That is why it is the simplest amino acid, because you need one N end and another C end, and these two cannot be linked to each other simultaneously. So the simplest thing that you can do is just introduce 1 carbon in between them. That is exactly what happens in glycine. All right. Now, what is the structure of serine? Serine is also easy. It's not difficult at all. You see, serine is also like COO minus, that carboxyl end is there. There is this carbon end also. The zwitterionic form we are writing. Now, serine is one of those amino acids which has a hydroxyl group. All right, so it has a hydroxyl group. So where would that hydroxyl group be? That hydroxyl group will be here with another carbon. This will be CH. So this is the structure of serine. All right. Now listen to me carefully. If we have to form from this to this, what do we need to replace, or rather what do we need to remove? We need to remove, if I'm not mistaken, this thing. All right, we need to remove this thing. So this is hydroxyl, and CH2, let's remove this as methyl. All right, so kind of what is happening is that you are transferring a hydroxyl group and a methyl group from something to something, and hence liberating serine of this thing, and hence forming glycine. Now, what are you going to transfer this thing to? This is where a very important molecule comes to play. The name of the molecule is tetrahydrofolic acid, THFA. Tetrahydrofolic acid, that is going to form N5, N10, methylene, THFA. That is tetrahydrofolic acid. What just happened here? Very easy. See, what I told you was you need someone to accept this thing, right? You need someone to accept this thing. That someone is tetrahydrofolic acid. Tetrahydrofolic acid is a very, very crucial molecule as far as 1 carbon metabolism is concerned. It is a very important compound which accepts 1 carbon groups, and methyl, although CH2 is not methyl, CH3 is, but this is a kind of a 1 carbon group. So THFA actually accepts this and converts into N5N10 methylene tetrahydrofolic acid. All right. Now, what will be the name of the enzyme here? The name of the enzyme is serine. All right. Hydroxy methyl transferase. So serine hydroxy methyl transferase. All right, that is the enzyme which is needed. Now, obviously I said that this is a bidirectional, so it's a reversible reaction as well. So if glycine, obviously in the presence of N5N10 methylene THFA, reacts in the presence of this enzyme, serine hydroxymethyl transferase, then it can form serine plus THFA. Just the reverse of this reaction. So you have to reverse this thing also. Obviously that's going to be the thing, right? Another very important thing which is I think worth to be noted is that this enzyme needs PLP as coenzyme. I think the name PLP should ring a bell because we have already studied about PLP, pyridoxal phosphate, when we had studied about transamination and oxidative deamination, right? So there also PLP was actually used as a coenzyme in that case also. So here also there is serine hydroxymethyl transferase. This enzyme uses PLP as a coenzyme. So this is the first reaction where glycine is formed from the amino acid serine. All right. Now, I said that serine forms glycine. So let's suppose that serine's brother threonine also forms glycine. Why did I say threonine to be serine's brother? Because both are hydroxyl containing amino acid. Easy, simple. That is why they are brothers. So one brother is forming glycine, the other one is also forming glycine. How? By the same mechanism? No. This has a different thing going on. Threonine also forms glycine, just as I said right now. The name of the enzyme is Threonine Aldolase. What is happening in this case? What is the structure of threonine? That we have to first, kind of understand. Now, why am I delving, or rather, why am I stressing upon the structures? Because they are not something which I am telling you to mug up. When we will be studying, or since we have already started with the specific amino acids and their metabolism, so the metabolic reactions will be very, very much easier if we kind of at least have something in mind about the structures. So I'm not telling you to mug up, like, draw a table and create, write up all the structures and then mug them up. No. With the reactions itself, we will kind of work out what the structures are. And they will actually help in the reactions as well. So it's a tit for tat. We are getting help from here also and there also. So what is the structure of threonine? Firstly, I have said that it is a hydroxyl containing amino acid. The second clue which I am giving you is the word 3. 3 means 3, obviously. Why does this ring a bell? This rings a bell because at the end, I know there is a methyl group. Interesting. Listen to me. Since threonine has 3 at the front, I know that CH3 is present at the end. Alright. So you need a carbon who has the hydroxyl group. You need a carbon who is holding the carboxyl end and the amino group. And that's it. You have threonine. So you don't need to memorize the structures. All you need to do is use some maybe mnemonics and use the reactions and connect them. Then they will be easier. So that's the structure, right? So rest we will be filling with the hydrogen atoms. Done. Now, if you need to convert this into this thing, what do you need to remove again? We need to remove this entire thing, kind of, right? How we are going to remove this thing? How are we going to remove this thing? Now listen to me. First, let me write the glycine part. That is CH2NH3. Alright? Plus, here there is no one to transfer it. See, we have not named our enzyme as transferase like the previous one we did. It is not transferase, right? It is aldolase. So it is not going to transfer the group. So it is going to be there. It's only splitting it. So what happens here is we get CH3, CHO, that is aldehyde, acetaldehyde. Alright, so glycine plus acetaldehyde. Right? So aldolase, aldehyde, rings a bell. See, So mnemonics are scattered all around. All we need to do is find them. So threonine aldolase is splitting this threonine, and we are getting obviously glycine here and acetaldehyde. And we know the structure of threonine and serine as well. So it's a double profit for us. Done? Done. Now we come to an extremely important, you can say, pathway for the formation of glycine. All right, what is that? That is, all right, let me demarcate. We'll use up this space. Now listen to me very carefully. When glycine is formed in our body, it has a metabolic pathway. As I said, it has many metabolic functions. Now, one of the very important metabolic pathways that glycine follows is known as the glycine cleavage process or the glycine cleavage system. In that, glycine is actually, broken down into carbon dioxide, ammonia, and also methylene tetrahydrofolic acid, which we had formed here, right? N5N10 methylene THFN. Yes, this thing. That is a one carbon group. So why am I telling you this? I'm telling you this because the glycine cleavage system is reversible. So if we can reverse the breakdown of glycine, we can actually form glycine, right? But for that, we need to understand how the breakdown works. So we need to understand how the glycine cleavage system works, right? Now, the glycine cleavage, this process is actually carried out by a multi-enzyme complex known as Glycine Synthesis enzyme. Glycine synthesis, when we are synthesizing glycine, or the glycine, we actually say glycine decarboxylase complex. Glycine decarboxylase complex or glycine synthase, right? So this is a multi-enzyme complex. Multi-enzyme complex means a variety of enzymes working together in a coordinated manner, different functions, but together they are actually carrying out something, and at the end you will get something. That is multi-enzyme complex. So what's happening is this multi-enzyme complex has different enzyme components, and we will discuss them one by one. At the very first, what happens is decarboxylation. A carbon dioxide is taken out. This is carried out by the enzyme PLP dependent glycine decarboxylase. PLP dependent glycine decarboxylase. Alright? PLP dependent glycine decarboxylase. This is the enzyme. Right? A carbon dioxide is given out. What remains is actually the amino methyl group, actually the methylamine group. So the methylamine group is shuttled, let me write it here first, methylamine group. This thing is shuttled to another enzyme, What that enzyme will do is two things. First, it will lead to THFA becoming methylene THFA. and second is liberation of ammonia. All right. Now this makes sense. Why? Because you had methylamine. So the methyl group is taking care of the THFA and the amine is liberated as ammonia. That makes sense. What is this enzyme? This enzyme is amino methyl transferase. So the amino methyl transferase. All right. Amino methyl transferase. I'm sorry, it's becoming a little congested. Maybe I should have taken a new page, but please bear with me. Amino methyl transferase is the name of this enzyme, which is leading to two things happening. One is THFA to methylene THFA. Second is the liberation of ammonia. Now, before I say what is going to happen next, I need to explain you just one more little thing. You see, these enzymes, they are proteins, right? Now, in a multi-enzyme complex, they are all linked together. These proteins are all linked together. And all these proteins, enzymes, they are held together, have a common link. That common link here is the what we say to be H protein. Again, listen to me carefully. This PLP dependent glycine decarboxylase enzyme, it is a protein. This is known as the P protein. That is why I wrote intentionally the PLP dependent glycine decarboxylase, so that you remember the P in the first. All right, that is the P protein. Done. The next one is amino methyl transferase. This is known as the T protein. The T protein is amino methyl transferase, right? What is the H protein that I was talking about? See, all these T protein, P protein, these are all held together. These are all shuttled by the H protein. What do I mean by shuttled? Is that this methylamine which is formed, it is formed in the P protein, because P protein leads to decarboxylation, removal of carbon dioxide. So the resultant is shuttled in through the H protein to the T protein. So the H protein is like a common pathway. It is holding all the subunits together. That is what the H protein is doing. Now this H protein has lipolic acid as its what we say to be prosthetic group. The lipolic acid is its prosthetic group, right? So when this methylamine or whatever the residue is after carbon dioxide has been removed from glycine, this actually is going on and binding with the lipolic acid, right? So what is happening is that the lipoamide residue of H protein, the lipoamide residue of H protein, that is actually reduced lipoamide. All right, that is what is happening. Again, I'm explaining. This thing happened, this residue, whatever it was actually linked to the H protein, to its prosthetic group lipolic acid, that is lipoamide. And in this process, when the H protein shuttled this group to the T protein, so that the T protein can lead to THFA becoming methylene THFA and the release of ammonia, in that process itself, this lipoamide became reduced. Alright. So if the prosthetic group is compromised, the enzyme cannot work. So this prosthetic group is recycled. How it is recycled is, this has become reduced. So it uses NAD plus and actually reduces the NAD plus into NADH plus H plus. Alright, NADH plus H plus. So this reduced lipoamide is again oxidized back to lipoamide, and NAD plus is reduced to NADH plus H plus. Hence, lipoamide is now restored, H protein is restored, H protein acts as a shuttle, and hence the next molecule of glycine can bind. Then again decarboxylation happens, the residue is shuttled by the H protein to THFA, to T protein, and this can go on, right? Now the enzyme responsible for this reaction here is lipoamide dehydrogenase. Alright, lipoamide dehydrogenase is what we say to be the L protein. So this is the other functional component of the glycine synthase, or rather glycine synthase enzyme, or glycine synthase multi-enzyme complex. That is, it has a L protein, H protein, T protein and P protein. And H protein is the center. It is shuttling it is coordinating and is leading to the coordination between the different other subunits. Right? Why am I telling you this? Does this lead to the synthesis of glycine? No. But then why is the name glycine synthase? Because this is reversible. So since we have studied how the glycine cleavage system works, if we reverse this, which we can because all these steps are reversible, so taking ammonia, this methylene THFA and the carbon dioxide, we can again form glycine. Hence, this is also an important way as to how glycine is formed. Alright, so this is also a very important way. The glycine cleavage system is very important because when we will be talking about the clinical implications of glycine metabolism, there we will see as to how the faulty glycine cleavage system leads to complications. Right. Now we will be changing the page at last. So now what we are going to do is we are going to start with the metabolic parts, or rather metabolic pathways which glycine is following. See, we talked about how glycine is formed, right? We talked about the different methods as to how glycine is formed, right? Now we will talk about how glycine is broken down and how glycine is forming different other products which are important for our body. The first thing that we talk about is related to our topic or our discussion at the very beginning of this video, that is the glucogenic pathway. The glucogenic pathway of glycine. So what is happening in that? What do I mean by this? At the very first, let me tell you that. See, we talked about glucogenic and ketogenic amino acids. So I said that all these amino acids, they are ultimately leading to the formation of one of those 7 molecules. So now we are going to see as to how glycine forms one of those 7 molecules, and hence finally leads to the formation of oxaloacetate and finally to glucose. Since it is glucogenic, we have already revealed it. So now the glucogenic pathway of glycine. The glucogenic pathway of glycine is what I can tell to be a little underwhelming. Why it is underwhelming is because glycine is converted to serine. You remember this very reaction, if you may. This was one of the steps, or rather one of the processes by which glycine was formed from serine. Now we are reversing the reaction and we are telling that serine is being formed by glycine. Why we are telling this is because it is serine whose glucogenic pathway is followed. It is serine which leads to the formation of one of those 7 molecules, not glycine itself. So that is why I said that it is a bit underwhelming. Glycine itself is not forming one of those things. Serine is. But glycine is converting to serine. Hence, glycine is glucogenic amino acid. So what is happening? Firstly, do me a favor and tell me which enzyme was this. Very good. This is serine hydroxymethyl transferase. And obviously there was THFA to, will there be THFA to N5-antimethylene THFA or the other way around? Yes, other way around. Why? Because we have reversed this thing. There we were studying about serine to glycine. Now we are studying glycine to serine. So you need a carbon donor. So that carbon donor is N5-methylene tetrahydrofolic acid, not methylene THFA directly, or rather THFA directly, I'm sorry. So after serine has been formed, what happens? What happens is dehydration happens. A water molecule is given out. So this is, the enzyme will be serine dehydratase, as simple as that, dehydratase. So serine dehydratase, this enzyme is used, water is given out, and what is formed? Amino acid is formed. How did this happen? If I tell you that from serine you get amino acid, and from amino acid you get pyruvate, this is kind of like mugging up the things. So let's go on. That is why I was telling that if you know a little bit about the molecular bits, just a little bit, you don't need to mug them up as much as you can manage while studying the metabolic pathways. Then it will be a lot easier to understand these reactions, and then the possibility of mugging up will also decrease. So we have already studied what the molecular structure of serine was, and what was that? Obviously you need a carboxyl thing, you need a amino thing, right? What other you need in serine is, yes, a hydroxyl group. Now hydroxyl group cannot be like directly linked to this carbon, so it needs another carbon, so CH2OH. This is what we say to be serine, and obviously there will be CH here. If you remove a molecule of water, what is formed is COO minus and C double bond NH. This is interesting. And this OH is given out. So in the form of CH3 it will remain. In the form of CH3 it will remain, right? So what's happening is that we are getting what we say to be from serine to amino acid. And how is that happening? That is happening because we are removing a molecule of water, and hence amino acid is being formed. Now what we need to understand is how from amino acid pyruvate is being formed. From amino acid, how is pyruvate being formed? You see, I forgot to write here in this step. It's not an instant thing from amino to pyruvate. Here one molecule of water is required, firstly. Secondly, a molecule of ammonia is given out. Interesting. What is happening here is that you are adding a molecule of water, so you just dehydrated it and now you're rehydrating it. But now the difference is that you are also deaminating it, right? So what is forming is pyruvate. How is that happening? Let's see. What is the molecular structure of pyruvate? Pyruvate is very important. It's too important to not know the molecular formula. So COO minus remains, right? The only difference is that C double bond NH, that is an amino group, has been replaced by double bond O and CH3. This is pyruvate. See, why the water was required was you had to replace this amino group in the form of ammonia, right? So I think the hydrogen donor in that case would have been water. I may be wrong because I'm not going into the details, because I don't know the details of the molecular rearrangement or how that is actually happening, the mechanism. But for the sake of simplicity, we can feel, or rather think, that water is actually donating its hydrogen atoms, not molecules, hydrogen atoms, and leading to the removal of the amino group in the form of ammonia from the reaction, and the oxygen itself is getting double bonded to the carbon and forming pyruvate. So that is an important pathway, and that is how the glucogenic pathway is occurring as far as glycine is concerned. As I've already told, if you form pyruvate, pyruvate will now form oxaloacetate by the reaction we say to be pyruvate carboxylase. So by pyruvate carboxylase, we will be getting oxaloacetate, and oxaloacetate by the process of gluconeogenesis will form glucose. Hence, from glycine to glucose, we can form that, and hence it is glucogenic pathway, right? So the glucogenic pathway is the first thing that I would have wanted to recommend. Now we go on to the other metabolic functions of glycine. What are the other metabolic functions which glycine has? Glycine has many metabolic functions. The first thing that we study now is very important. Its importance cannot be stressed enough. That is creatine and creatine phosphate formation. What is this all about? Everybody knows about creatine and creatine phosphate, right? They are used in the muscles in the form of energy donors, right? We all know this. Now, glycine is used in the formation of creatine, and incidentally creatine phosphate also. Hence, we are studying their formation under glycine metabolism, right? The first thing that we need to remember is that 3 amino acids make up creatine. What are those 3 amino acids? First one is obviously glycine. Second one is arginine. And third one is methionine. Alright, so glycine, arginine, methionine. These 3 amino acids together make up creatine. Now, how that is actually occurring? Let's see. See, arginine, in this pathway of creatine creation, of the formation of creatine, I'm not going to go into the molecular basis of this, alright, because I think that would be too much. So since it would be too much, so I would like to kind of cut to the chase here. Glycine, alright, at the beginning it reacts with arginine. At the beginning it reacts with arginine. Now, arginine has a group. What group is that? It is C double bond NH, NH2. I would like you to contrast this group with another group, C double bonded O NH2. Does this ring a bell? It should. CONH2 is amide group. So we are replacing this oxygen with NH, that is nitrogen. So what will this group be called? Something close to amide but not yet close. What is that? Amidino group. The amidino group is what this is called, right? So since it's quite close to what we say to be amide, so this is known as the amidino group, right? Now amidino group is present where? Present in arginine. Arginine transfers that amidino group to glycine, resulting in the formation of what we say to be a very interesting name, guanidoacetic acid. All right, guanidoacetic acid. That is the name of the product which is formed. That is glycine after the getting the amidino group, it is forming the guanidoacetic acid. Now arginine after losing its amidino group becomes ornithine. Yes, the famous ornithine of urea cycle. So it becomes ornithine. So see, we don't have a problem here because ornithine is quite famous, so it's quite easy to remember ornithine, right? And glycine, arginine, these are also famous amino acids. Guanidoacetic acid is a bit tricky name. I'm going to give it that. So it has a novelty in terms of name. So do me a favor and remember this. All right, can you do that? I hope you can. So guanidoacetic acid is after glycine has got the amidino group from arginine, and arginine after losing it has become ornithine. What happens next? The next step that actually occurs is very direct. That is, methionine comes into play. Now methionine cannot just go and react with guanidoacetic acid. There has to be some other larger group which donates methionine. What is the primary, I would say, methyl donor in our body, or methionine donor if you may? It is S-adenosine methionine. S-adenosine methionine. All right. So I'm abbreviating that here, SAM, S-adenosine methionine, that donates the methyl group to guanidoacetic acid and itself gets converted to what we say to be S-adenosine homocysteine. All right, S-adenosine methionine to S-adenosine homocysteine after it has donated the methyl group, and guanidoacetic acid after reacting with that forms our product which is desired, that is creatine. So creatine is formed after guanidoacetic acid reacts with what is donated by S-adenosine methionine, which itself forms S-adenosine homocysteine. Now we have formed creatine. Our aim is not only to form creatine but creatine phosphate as well. So how will creatine phosphate be created? That is also simple. You just phosphorylate creatine. Simple. So who is going to donate the phosphate group? ATPs. So ATP to ADP, this will actually occur. And by the way, I actually forgot to mention the name of the enzyme here. So what will the name of the enzyme here be? You're actually transferring something, so it will be a transferase, right? So the name will be acetic acid, you can abbreviate that as acetate. So guanidoacetate, and what are you donating here? Methyl. So guanidoacetate methyl transferase. So let me write that, it's quite a big name. Guanidoacetate methyl transferase. All right. So quite logical name, big name, but logical, I would create it. So creatine is formed. Now phosphorylation takes place and creatine, phosphate is formed. Creatine phosphate is formed. This phosphorylation is taking place here, right? Now, what is the name of the enzyme which is responsible for this? That is creatine kinase. So creatine kinase is the enzyme which is responsible for the formation of creatine phosphate from creatine due to phosphorylation by ATP, forming ADP. Now, how exactly does creatine phosphate provide energy? You see, creatine phosphate is nothing but a phosphate holder. So it will donate the phosphate to ADP, forming ATP, hence reversing this step here, forming creatine again. Alright, and then the ATP can be used for the energy. So creatine phosphate itself does not break down and form energy like that. It's not like that. It is actually kind of a phosphate, I would say, donor. It will donate its phosphate to ADP to form ATP, and hence the ATP can be utilized for energy, right? So this what I would say, reaction of creatine phosphate back to creatine by the same thing, except you are actually reversing ADP to ATP. This is called the Lohmann's reaction. Lohmann's reaction. Alright, so this is a very important thing because now ATP can be utilized for energy, right? Now you may have heard of the term creatinine as well, right? So what is the difference between creatine and creatinine? People used to get confused with these two terms, that is creatine and creatinine. So we need to clear up that confusion right now. Creatinine is the dehydrated form of creatine, and creatinine is actually a waste product as well. Alright, so this creatine phosphate, it actually can be dehydrated, right, to form creatinine. Oh, sorry, I kind of so creatinine, right? This reaction does not require an enzyme. This is very important to be noted because this is a non-enzymatic spontaneous reaction. So this is spontaneous reaction. So this creatinine, this is what? This is actually a excretory product. Excretory product. That is this creatinine. Right. So that is what the creatine metabolism is all about. And obviously I forgot to give something here. If creatine phosphate is happening, then you need to dehydrate, so water will be removed, and phosphate is also needed to be removed. Inorganic phosphate is also being removed. So water is removed, inorganic phosphate is also being removed. So that is also being done. So this is what I would say to be all the very detailed and important metabolic reactions which glycine goes on. There are many other functions of glycine, and I'm going to just skim through them very quickly. I'm going to skim through them. then we can go on to the disorders and complete this. All right. So just to name some of the other things that glycine is actually involved in is, firstly, heme synthesis. Heme synthesis. You know, oh, sorry. Camera is actually disturbing today. So in heme synthesis, glycine is actually the starting material. Glycine and succinyl-CoA, they react and they form delta aminolevulinic acid, and from there a variety of steps actually occur, and finally we get heme. That we study under heme metabolism, because we actually have that in as far as the scope of biochemistry is concerned. So synthesis of heme is an important obviously function which glycine has. Right. Another very important function is purine synthesis. This is again going to be dealt when we study nucleotide metabolism. So purine synthesis is also an important function of glycine, and glycine is actually incorporated in the ring of purine. We'll see to that, don't worry. A very important function of glycine other than these are something which I've already discussed, that is glutathione formation. If you remember, glutathione formation. When I had talked about the absorption of amino acids, I had talked about the Meister cycle. There I had talked about how glutathione is formed. So glutathione is actually what we say to be gamma glutamyl cystinyl glycine. So there are three amino acids which are actually forming glutathione. They are obviously glutamate or glutamic acid, cysteine, and glycine. So glutathione formation is also a very important function of glycine, since glutathione is a very important antioxidant in our body, and it protects our body cells from all kinds of destructive superoxides and peroxides which will rupture the membrane and lead to damage. So being an antioxidant, it's a very important molecule for our body, and glycine is one of its constituents. All right. Now glycine is also used as a neurotransmitter in our body. I'm just listing these things, all right, since we have already discussed the very important functions which needed detailed elaboration. We have already discussed those, so I feel like these can be schemed through. So it can be used as a neurotransmitter, in fact, and it actually, what kind of channels does this open? It opens chloride specific channels in our body. These are just information which I'm listing out. So chloride specific channels actually opened by glycine, which is used as a neurotransmitter. Now another important function of glycine is its function as role as a conjugating agent. Now, what does that mean? Conjugating, yeah, I got the spelling right, agent. It means, if you remember bile acids, do you remember bile acids and bile salts? Sodium glycocholate, taurocolate, etc. So glycolic acid, right, that is actually what we can say, that glycine is actually used to conjugate these bile acids to produce bile salts. So it is a very important conjugating agent. So what are the bile acids which are the main conjugated bile acids, we can say? Examples of those, they are glycolic acid, and another very important name is the glycosaminoglycolic. The glycosinodeoxycholic. So glycolic and the glycosinodeoxycholic acid, These are important bile acids and they are conjugated by glycine. Another example is not that of the bile acids but that of benzoic acid. Now this is important, hear me out. When I say this is important, it basically means it has a clinical correlation, because obviously, as I have said this 1,000 times before in my videos, that this is a professional course and whatever you study has got to have some clinical correlation. So benzoic acid actually is conjugated with glycine to form a very important compound known as hippuric acid. Hippuric acid. Hippuric acid is nothing but benzoyl glycine. Benzoyl glycine. Right? Now, why I said this is important is because this reaction, this conjugation occurs in the liver. Alright? This occurs in the liver. Why this occurs is because as a detoxification of benzoic acid. So since this occurs in the liver, so their amounts can be used as an index for liver function. So in LFTs they can be used in liver function tests. This reaction can be used because this conjugation reaction is happening in the liver, and the different levels of these acids will give us an index as to if the liver is functioning properly. That is the liver function test. That is why it's having a clinical correlation, and hence I feel like this is important. Benzoic acid plus glycine, this conjugation is giving hippuric acid. Right? Now obviously glycine is an amino acid, so amino acids are building blocks of proteins. So glycine is obviously constituents of proteins as well, right? So that is something which is obviously it goes without discussing as to how important and constituent of different proteins it is. Like in collagen it can be present in many other proteins. So I feel like I don't need to discuss those. Do me a favor and go through your book, and obviously you will understand glycine as a constituent of proteins. I'm here to discuss the more important points here, so please bear with me. Right now, I feel like we have done quite the justice to glycine as its metabolic pathways is concerned. Now I feel like we should go on and kind of discuss the metabolic errors in relation to glycine. The metabolic errors in relation to glycine. Remember what I had told you in when I was discussing about the glycine cleavage system? I had said that a faulty glycine cleavage system can lead to a problem. This problem is known as non-ketotic, sorry, let me get this a bit in angle, yes. So non-ketotic hyperglycinemia. Alright, so non-ketotic hyperglycinemia. So what happens in this non-ketotic hyperglycinemia? As I've already hinted, that it is something related to a defect in the glycine cleavage system. Alright, so due to some enzymatic defect in the glycine synthase enzyme, multi-enzyme, what would happen? The cleavage would not take place, the metabolic breakdown would not happen, so glycine levels would increase in blood, right? So this increase is not only in blood but also in other body fluids like urine or cerebrospinal fluid. These are all increased glycine containing body fluids. Alright, what are the symptoms? What do we see due to this defect? What happens due to glycine increasing in our blood? Well, firstly, what happens is osmolarity increases. Since we are talking about body fluids here, so osmolarity of the body fluids is increasing. Hence, this is also known as hyperosmolaric non-ketotic hyperglycinemia. So, it is also abbreviated as HONK, hyperosmolaric non-ketotic hyperglycinemia, right? So there are many clinical cases which we would have to see when we reach the professional level in our house staffship days. We will have to see this kind of HONK cases, that is hyperosmolaric non-ketotic hyperglycinemia. So the first thing that happens, as I said, that the osmolarity of the fluids increases. Now, what is the problem? The problem is mental retardation. Mental retardation, all right? Mental retardation is seen. Seizures are also seen, right? These are all symptoms related to HONK, right? Now, the thing is, what do we do if somebody has a HONK? What do we do then? Problem is that the defect is in the enzyme. So it is basically a genetic defect. So genetic counseling is needed. But unfortunately, the bad news is that there is no effective treatment for this kind of a condition. All right. So we will have to bear with this and kind of act for the temporary cure that we can find for the alleviated, mental retardation, counseling, and to decrease the seizures. We have to work on that. But as a whole, since the problem lies in the genetic defect, so that's why it's very problematic and there is no effective management for this. Now we move on to the next defect, and that is the last defect, and that is also the most important defect, or rather metabolic error related to glycine. That is primary hyperoxaluria, right? Primary hyperoxaluria. What is this? You see, when I had talked about glycine's metabolic pathway, or the different metabolic steps that it actually follows, I had not talked about glycine's conversion to glyoxalic acid. What is all that about? Alright, let me tell you. You see, you remember transamination reaction? Glyoxalic acid is what I like to think of as the alpha keto acid of glycine. All amino acids, after being transaminated, they form a corresponding alpha keto acid. So glycine also kind of forms a transaminated product, which is glyoxalic acid. So for transamination to take place, you need another pair. In this case, the other pair is pyruvate to alanine. Alright, right? So the name of the enzyme will be glyoxalate, or rather alanine glyoxalate aminotransferase. So alanine glyoxalate aminotransferase. Done. Now, the thing is, this glyoxalic acid, what is its structure like? Glycine was something like COOH, right? After transaminating, what you do is COOH, you remove this and you introduce a double bond group. That is what you do in transamination. So if you remove that, you also have to remove a hydrogen here, so CH. So COOH, CHO. Now if this is actually reduced, okay, let me write the name of the product here and let me write the molecular structure here. So if this is an aldehyde group, so if an aldehyde is reduced, what does it form? It forms alcohol. So CHO becomes CH2OH, and the name of this is what we say to be glycolate, right? And obviously NADPH to NADP plus is happening here. Why this is happening? Because you're reducing glyoxalic acid to glycolate, right? And obviously this is known as glyoxalate reductase, right? Now what can happen is glycolate is oxidized to, that is, oxalate or oxalic acid. Oh, I'm so sorry. I didn't see the camera. I'm sorry. Here's the thing. All right. So glycolate can form oxalate. Also, you can form glyoxalic acid to oxalate directly by oxidation. All right. Why am I actually showing all this? It is because this primary hyperoxaluria is actually accumulation of this oxalate in our body due to accumulation of this glyoxalic acid. Why does that happen? That happens either due to, number one, defect in this enzyme, that is alanine glyoxalate aminotransferase, or number two, defect in this enzyme, that is glyoxalate reductase. Defect in these enzymes leads to an increased pool of this glyoxalic acid in our body, and hence more amount of glyoxalic acid means more amount of oxalate is formed. This oxalate is actually deposited in the kidneys as kidney stones, right? And hence the condition is known as nephrolithiasis. Nephrolithiasis and also urolithiasis, right? So kidney stone, this is a very common thing that we hear. This is due to accumulation of oxalate stones in the kidneys, right? That is due to this. Now, if it is due to defect in this enzyme, we say it is primary hyperoxaluria type 1. But if it is defect in glyoxalate reductase enzyme, we say it is primary hyperoxaluria type 2. In type 1, what we have is kidney stones and nephrolithiasis, obviously, and obviously there is also hematuria. Hematuria means there is blood in the urine. But in type 2, we have urolithiasis, and it is milder, right? So what do we do to combat this? To combat this, you increase your water intake of water, because more amount of water, so there is the possibility of washing away these oxalates, and you reduce the dietary oxalate intake. Now, something which I did not mention previously, which I should have mentioned, is why is this defect in alanine glyoxalate aminotransferase seen, or what is the defect? Why is this enzyme being inactivated? This is a very important concept and I actually missed it, so I'm very sorry. This is actually what we say to be a protein targeting defect. A protein targeting defect. What does that mean? This enzyme, alanine glyoxalate aminotransferase, this is actually a peroxisomal enzyme. So it is supposed to be present in the peroxisomes. Which organ, by the way? Where is all this taking place? This is actually taking place in the liver. So all of this is actually taking place in the liver, and in the liver hepatocytes in the peroxisomes. That is where this should take place. But due to protein targeting defect, this hepatic peroxisomal enzyme, alanine glyoxalate aminotransferase, instead of being transferred to the peroxisome, it is transferred to mitochondria. So in the mitochondria, a peroxisomal enzyme cannot work, right? So it is inactivated. So hence a protein targeting defect actually makes this enzyme inactive, and also this enzyme also, and hence there is primary hyperoxaluria because an increased pool of glyoxalic acid is formed. Hence oxalate is formed in large amounts and it deposits as kidney stones, leading to nephrolithiasis, urolithiasis, and so the management of this condition would be how? By increasing the water intake in our body, so that the oxalates can be excreted out. More and more amount of water means there is increased chance of excretion of these oxalates. And secondly is that you already have a lot of oxalate in the body, so you don't want any more of that. So you decrease dietary oxalate so that external source of oxalate is compromised. Now there are a list of foods where oxalates are present in high amounts, and that is like tea is there, cocoa is there, leafy vegetables, are many leafy vegetables present a large number of oxalates. So that there is a whole list of that. Those foods need to be actually avoided so that oxalate amount does not increase any more. So that is all about primary hyperoxaluria, and that is also all about the metabolic errors in relation to glycine, right? So next time we'll be starting with the next amino acids. Today we studied about the concept of glucogenic and ketogenic amino acids, and we also studied about the metabolism of the amino acid glycine as a whole. So in the next video we'll be seeing about the different other amino acids and their metabolism, right?